Okay, hello. Thanks for having us. We're excited to introduce Multis to you all. So Multis is a team of medical and doctoral students, and we came together with the goal of innovating diagnostics designed for global health. So we decided to innovate diagnostics for neglected tropical diseases. Neglected tropical diseases are a diverse group of infectious diseases that have largely been overlooked. They infect more than 1 billion people worldwide, and every single year they cause more than 500,000 deaths. However, they also cause a significant amount of suffering, and they can have a disabling impact on people's lives. For this reason, the CDC calculates that 57 million years of life are lost annually due to premature disability and death. That's roughly equivalent to the burden of disease of tuberculosis. Thankfully, treatments for neglected tropical diseases exist, but diagnostic tools that can reach people with NTDs are limited. So why do we need better diagnostic tools for NTDs? There are a few reasons. First, NTDs affect the most impoverished people in the world, people who often lack access to hospitals. Therefore, we need diagnostics that can reach people in the field and provide rapid results. Second, many diagnostic tools required cold storage from their point of manufacture until they reach the patient. That's otherwise known as the cold chain. In order to reach patients with NTDs, we need to break the cold chain and provide temperature-resistant diagnostic tools. Third, the WHO, USAID, and many other global health organizations have made significant progress already in reducing the rates of NTDs. However, they have all repeatedly called for a need for more sensitive diagnostic tools to monitor surveillance. And finally, I want you to remember this fact. All low-income countries are impacted by at least 5 NTDs simultaneously, and often the same person can be infected by more than one NTD at a time.
To address this need, we designed a diagnostics platform that's capable of screening for multiple NTDs in one low-cost test. Here's how it works. You take a blood sample, you introduce it into the Multis array, which is a microfluidic chip that handles the biochemistry for the panel of tests. The chip is inserted into a device that quantifies and displays the results as cheaply and intuitively as possible. And the device stores the results internally and geotags them so they can later be synced to a computer or smartphone so the data can be sent to the cloud and tracked by the WHO and local ministries of health. We prototyped this here in the Mount Sinai Rapid Prototyping Center. Getting this to work required some mechanical design, electronics, software, and biochemistry. So it's been really helpful that our team has a diverse set of talents. And at the core of the device is a type of molecule called an aptamer. These are specifically DNA aptamers. The great thing about them is they're really cheap. They're very temperature resistant, so they can be used without a cold chain. And yet they can be designed to bind to things with high affinity and specificity. They're really easy to synthesize and design. And in fact, there are even published aptamers for specific neglected tropical diseases that we can leverage.
Taking a look at the competitive landscape, some but not all NTDs have diagnostic tests, and only a few of these tests are actually usable in the field. There are also only testing for one disease at a time and are subjected to subjective interpretation, which can lead to high rates of false positives and false negatives. However, the gold standard for diagnosis of many NTDs remains clinical exams or lab assays, and these require expensive equipment, trained specialists and doctors, as well as access to a healthcare system. And so Multis is working on developing field-ready technology that can diagnose multiple diseases at once, which is a major advantage compared to what's currently available. We believe we have a strong competitive advantage over current rapid diagnostic tests. The World Health Organization provides a list called the Assured criteria, which they propose are essential for successful rapid diagnostics. In accordance with these criteria, our technology is affordable. It uses biotechnology that is well-developed and inexpensive. It's sensitive and specific enough for global surveillance programs. It's user-friendly with a simple user interface and rapid, objective outputs. It's robust in harsh climates and does not require cold chain. It's equipment-free, meaning it's portable and doesn't require any expensive equipment or lab bench, and it will be easily delivered to those who need it. And in addition to these criteria, our device will also be able to detect multiple diseases in one, and this is the major advantage that we hold. We calculated our market size based on the total number of people at risk for NTD infection globally, which is approximately 2 billion people. The average cost of a rapid diagnostic test for an NTD is about $1.50. Surveillance programs typically require a minimum of 3 years of disease testing, and using the estimate from the CDC that low-income countries are affected by at least 5 NTDs simultaneously, the total market for rapid diagnostic tests for NTDs is a large number, about $40 billion US. Our first iteration of this product will detect disease in sub-Saharan Africa, and that narrows down our market to about 23 billion. And if we assume that we're going to capture 5% of this market, our serviceable obtainable market is a little bit over $1 billion.
So how are we going to do this? We're currently designing the two components of the platform, the detection device and the Maltus array, to be functionally tied. So that's something that you'll see in the razor blade model and the inkjet printer. So we want to leverage economies of scale, so the high volume, low cost nature of consumable purchases to cover the cost of the actual detection device. And we're ultimately doing this because we want to offer the device completely for free. Who are we targeting? So you can imagine the big players in the space, nonprofits, USAID, the WHO, the Gates Foundation. The pharmaceutical industry is donating about $1 billion worth of drugs a year, but we're actually looking to target local governments, not just because they feel the largest economic burden per individual of per infected individual, but because their involvement is crucial for the long-term sustainable solution of eradicating these diseases. So where are we right now? We have a functional prototype that we're using. We're testing different versions of our chip to find the one that is most scalable and cheap for manufacturing. We're also looking to raise some money, so we're turning to friends and family as well as CrowdRise, a CrowdRise campaign to raise about $10,000. And with this money, we want to expand our disease testing as well as refine the design of our device and chip. From there, we'll kick off software development as well as establish partnerships with academia and industry to get this into the field faster. But looking ahead, our device, the underlying biology and technology, is very well suited for scalable growth. So right now, we're targeting 5 NTDs in a specific location, sub-Saharan Africa, where the need is most critical and crucial. But we want to take aptamers that are already published in literature and expand our array offering by region, so NTDs that are prevalent in different areas of the world, as well as target larger market diseases such as malaria, TB, HIV. In the long term, we also recognize that this technology is not just limited to diseases or developing markets. It can also be applied to developed markets like here in the US. You can imagine that you can expand the Maltus array offering, right, as a community health device that can scan a patient's sample and assess their general health. So here's a quick look at our team. As Mika mentioned, we're a group of Sinai doctoral and medical students. We came together about a year ago. We bring together really diverse backgrounds: math, immunology, computer science, and economics. And we were all really driven about having an immediate impact in public health. I'd like to highlight the tall one in the team, Brendan, who is here with us today, but he couldn't present with us because he's actually defending his thesis in 2 days. So on that note, thank you very much for listening.
Do you have any data, even if it's very, very early, to show us that and I understand you're in the process of actually building the prototype, but do you have any data to show that the microfluidic technology can allow a sensitive and specific detection of any of the 5? Doesn't matter if you can combine them right now, but just individually, do you have any data to show sensitivity or specificity of the detection?
So right now, we've only tested it on test biomarkers, not actual tropical diseases. And so that's why the money that we're trying to raise might seem kind of absurdly small, only $10,000, but that's the money we think we need to basically get that data that you're talking about and finish the prototype to the point where we're confident that we can actually detect the diseases with a reasonable volume of sample, and then we would look forward after that.
So, now I'm struggling. So, is there an underlying technology that already exists, or are you building it from scratch? Or now
So we're integrating things. So the underlying technology is really aptamers. That's the molecular side of detecting
So do you have
things.
are there any data showing that aptamers are used in a detection device, in a molecular diagnostics device?
Yeah,
at all?
actually, so
That you could use that to give us a confidence that the underlying technology works.
sure. So the interesting thing about that question is that aptamers have been tied up in patents for a long time, patents that recently expired. And so for diagnostics, they were exclusively licensed to a company called SomaLogic, which has been working on such things, but it's held it sort of only in one place. And only in the last couple of years has this been freed up. So SomaLogic has some interesting technology. They've gone in a different direction. They're looking at biomarker discoveries for the US. But they certainly have made a lot of interesting achievements.
A lot of diagnostics companies have spent a lot of time and a lot of money on microfluidics and sample prep. Is there someone you're piggybacking on for that technology, or that's something you need to do in the future?
I mean, I think as we go through more experimentation, that's something that we'll look into to really assess what do we need. But what we know right now in literature is that aptamers are extremely good at detecting the biomarker in extremely low concentrations without much sample prep. So we really hope that it's just a very easy segment into the market such that we don't need any upstream processing before actual detection.
Part of your challenge is blood is a very complex fluid to analyze.
Correct. Again, another thing that we'd have to address during testing, but another advantage of aptamers is extremely specific for their targets. I don't want to claim too much about it, but this is all that we've pulled from literature and our understanding and our experiments that we've done already in blood.
To what extent have you guys engaged with either, for example, FINE in Geneva or any of the sort of end users on shaping your product profile? For example, when you quote the $1.54 price for the test, where is that coming from?
So the second question, the $1.54 is a weighted average of the current rapid diagnostics that are available in the field for these NTDs. So we wanted to use that as a baseline. In terms of how much interaction have we had with the organizations that are involved with these diseases, it's minimal up to now because we really want to make sure that we have a solid product and solid biology that we know is ready for the field. So we want to go to the Gates Foundation, for example, for a larger sum of money where we know we're ready to really enter the field for testing.
Okay.
How are the specific NTDs chosen?
So I actually predicted you would ask that question, and this is something that we've been thinking about for a while. So I have some slides. The WHO lists 17 diseases as NTDs, so we decided to focus on the ones in red, and there's a reason for that. So neglected tropical diseases have been the interventions for neglected tropical diseases in public health, you could pretty much divide them in two. There are the diseases that were targeted by mass drug administration, so you just give a drug to an entire community, like an antiparasitic, to address that disease, or there are the diseases where you need to find the individual patient and treat them individually. The WHO, USAID, and the Gates Foundation have specifically called for a need for better, more sensitive diagnostic tools for surveillance, so in order to monitor those mass drug administration programs. And that's why we chose those 5 diseases: foodborne, trematoid, infectious, lymphatic filariasis, river blindness, schistosomiasis, and soil-transmitted helminths.
Has the primary problem been cost, or has it been something else?
So why has there been a lack of innovation
Yeah.
sort of in the diagnostic market? part of it is cost. A second part of it is that as you go through the timeline of surveillance, we've thought about this a lot. As you go through the timeline of surveillance, initially, cheap, rough, rapid diagnostic tools were sufficient to get a baseline for mapping, right? Just a baseline prevalence rate so that you could know where to target those drugs. However, as the prevalence of these diseases has dropped, we need more sensitive diagnostic tools. So this is really the first time there's a specific need for a very sensitive diagnostic tool in neglected tropical diseases.
Yeah.
And additionally, with mass drug administration, there's now the concern of resistance and co-infection with other diseases. So that's the exact entry point for Multis, because this is an area where you're gonna need a diagnostic tool that tests for multiple diseases at the same time, because it not only impacts the treatment, it totally impacts the mass drug administration as well.
So perhaps an analogous situation is if you think of Cepheid and MDR-TB and the setup of the surveillance networks for TB, why wouldn't the systems go to something like that, a little bit more centralized?
We wanted to focus on neglected tropical diseases, but ultimately, if we did want to turn this into a single disease test, there is an argument to having a multiplex assay no matter what, because you could add multiple biomarkers for the same disease on that test and therefore get an overall more sensitive result for that specific disease.
Can I ask a question about, you guys are at least one of you is a budding medical doctor, and I spent a lot of time drawing bottles of blood from patients to look for bacterial infections. How do you find these organisms in a single drop of blood? It seems like the volume that you're assessing, even if you have a highly sensitive and specific, are going to catch an organism, especially large organisms like parasite, in a single drop of blood.
So I'll try to tackle that question. There's still a lot of questions that are surrounding exactly what volume we do need, but we are going off, again, our experience with experimentation as well as what is heavily cited in literature as very small samples of patient blood are actually needed to detect in the nanomolar concentration of the biomarker in the blood. And that's even quoted for specific NTDs that they've tested with aptamers. So a lot of it is based on what we've read in literature, but we'd like to back that up with further experiments.
And I just want to add that our goal is not necessarily to do this with a very small volume of blood. That would be nice, but the goal is to get the diagnostics done. So whatever volume is required is what we would use for that test.